Okay, thank you, Keith, and for those two really engaging sessions, and thank you, Lenny, for being with us. These are fascinating areas. They're of increasing interest to our students and postdocs, and who care more maybe than any generation past about the ethical dimensions of scientific research and the work that we do in the laboratory, and I appreciate that interest. So it's my pleasure now to introduce Professor Corinna Darian-Smith to present the annual Sammy Kuo Awards. This is a particularly celebratory time of our annual symposium where we recognize very high quality hard work and publications from the previous year by our graduate students and postdocs. And I'll let Corinna tell you about the award and its purpose. And Corinna's joining us virtually up on the screen behind me, and I will be Corinna's lovely assistant, and I will hand out the awards here. But just a personal note before I turn the floor over to Corinna. Corinna just was successfully promoted to full professor here at Stanford. So congratulations, Corinna.
Thank you.
And take it away.
Okay, so I have the great delight to introduce each of the recipients of the award this year. And I just want to start by giving a little bit of background to the Sammy Kuo Neuroscience Award. Sammy Kuo was born in Taipei, Taiwan in 1984 to parents Yao Qi and Liling Kuo. And in 1996, when he was just 12 years old, he was diagnosed with a rare inflammatory disease, which arose from the measles virus. His family cared for him at home until he passed away at 22 in 2006. And after his death, his cousins Peggy and Amy, who I'm delighted to say are both here at the meeting virtually, they were both graduates at Stanford. They contacted the Neurosciences Institute at the time, and the Sammy Kuo Memorial Scholarship Fund was established to support research in neuroscience. The opportunity allowed the family to memorialize their beloved Sammy and recognizes crucial neuroscience research that may one day help cure other children with debilitating conditions. The award has become truly prestigious over the years. And though born of sad beginnings, it's very much a celebration, as Bill mentioned, and it highlights the great importance of doing basic neuroscience research that recognizes exceptional early career papers. All of this information is on the website, the Sammy Kuo website. We're now in our 14th year, and the standard has become extraordinary. You can just see some of the recipients in previous years here, and these are just the first place awardees. And before I get going, I just want to thank the selection committees, the postdoc selection committee and the grad student selection committee for all their work behind the scenes to make this happen. And I'd also like to give a shout out to Elise Kleeman, who's done an enormous amount to bring this, to make this possible. So let's get started. We start with the Graduate Students Awards, and an honourable mention this year goes to Kevin Guttenplan from the Gittler Lab for his beautiful paper in Nature. Kevin and colleagues set out to identify the key protein responsible for astrocytic neurotoxicity, which is responsible for the active killing of neurons in certain neurodegenerative diseases, and discovered surprisingly that the factor involved was not a protein but a lipid. The work combined biochemistry with genetic screening approaches for receptors in neurons and secreted proteins in astrocytes using CRISPR libraries. They show that by eliminating the formation of the lipids, astrocyte-mediated toxicity was mitigated in vitro and in vivo. And the work suggests a new mechanism by which astrocytes kill cells in the CNS and has far-reaching therapeutic potential. And I'm hoping that everyone, yes, I can see Kevin there. Everyone who's up on the screen has to be visible online for everyone. Our third place grad prize this year goes to two people, Sam Vesuma and Isaac Cueva from the Daiseroff Lab, for their beautiful paper in Nature. The paper, which was highlighted on the journal's cover, describes a fascinating set of discoveries about the neurophysiological basis of dissociation, a psychological state in which the integrity of the conscious experience is disrupted, and there can be a dissociation between the sensory stimuli and responses that normally accompany that stimuli. Prior to this work, its neural basis was completely unknown. They used and even developed a wide variety of cutting-edge neuroscience tools, as we might expect from this lab, to identify a molecular, cellular, and neurophysiological basis for dissociation, and then connected this with physiological recordings in an epilepsy patient experiencing self-reported dissociation. The work is really pretty spectacular and is of major basic and clinical importance. Isabelle Lau from the Giacomo Lab is this year's second place finalist for the grad component of the Sammy Kuo Award for her paper in Neuron. Isabelle wanted to understand how a high-order cortical region, the medial entorhinal cortex, generates flexible neural representations capable of supporting navigation and spatial memory. She used newly developed neuropixel recording probes to record from hundreds of neurons simultaneously across the medial entorhinal cortex. And she did this over long periods of time while tracking rapid changes in the neural activity. She combined this with virtual reality and was able to show with her collaborators that the entorhinal circuitry can rapidly and reversibly transition between two or more navigational coding schemes without any change in the sensory environment. The work is enormously important for our understanding of vertebrate cortical flexibility, and it paves the way for many future studies of this at a network level. So this year's grad student paper of the year goes to Eddie Alberin from the Ding and Schatz labs for his article in Neuron. Dendritic spine dynamics are thought to be substrates for motor learning and memory, and altered spine dynamics often lead to impaired performance. So in this truly beautiful and collaborative study, Eddie and his colleagues discovered that there are cortical limits on motor performance in adult mice that can be lifted when a particular receptor, known as paired immunoglobulin receptor B, or PER-B, which is expressed in cortical pyramidal neurons, is knocked out or blocked. So PER-B knockout mice are able to learn a skilled reaching task much faster than their wild-type litter mates, and they nicknamed these the Olympian mice for obvious reasons. The work provides key evidence that increasing the stability of newly formed spines is sufficient to enhance motor learning, and artificial increases in spine stability during learning seem to correlate with enhanced performance. Since the limits on motor learning can be lifted by manipulating PER-B, even in adulthood and even in the healthy brain, the work suggests that blockade of this receptor may prevent spine instability and the accelerated loss of spines observed in various neurological and psychiatric disorders. Beautiful work. So we now turn to the postdoctoral awardees, and an honourable mention goes this year to Jinye Day from the Sudoff lab. And excuse me if I butcher people's names, it's my accent. Jinye was interested in understanding the molecular rules of synapse formation. And to do this, she studied the mechanism by which alternative splicing of neurexins regulates the properties of synapses. The work is truly pioneering as it delineates a surprisingly economical molecular mechanism by which neurexin signaling controls AMPA and NMDA receptor responses in synaptic circuits. Precisely assembled. Daniel Pederick from the Low Lab receives third place this year for his paper in Science. The paper addresses a key question in brain wiring: how are parallel networks precisely assembled to ensure that axons select targets in appropriate subregions? Though much is known about axon guidance in invertebrates and simpler vertebrate circuits, far less is known about the mechanisms of target selection in complex neural and parallel networks networks of the mammalian brain. Daniel showed that there was a complementary expression and reciprocal repulsion of two cell surface proteins involved in segregating the medial and lateral hippocampal networks. He also found reciprocal expressions of these proteins beyond the hippocampal network. Such multifunctionality of wiring molecules and their repeated use contribute to how a limited number of cell surface proteins can specify diverse connections in the mammalian brain. Project used new genomics. This year's second place goes to Maya Mayo-Nof from the Gittler lab for her paper published in Cell. Maya's project used new genomics technologies to make important new discoveries about mechanisms of the neurodegenerative disease ALS. Mutations in the C9orf72 gene are the most common cause of the motor neuron disease ALS, but it's not clear how these mutations cause the disease. And so Maya discovered that mutations in this gene activate a remarkably specific transcriptional program in neurons, converging on a single transcription factor, P53. She extended her in vitro findings to an in vivo model and showed that P53 knockout mice live at least 2.5 times longer. She even validated her findings in human patient samples by showing that P53 reduction is sufficient to rescue derived motor neurons from DNA damage. The paper really represents a major advance in the neurodegeneration field. And this year, first place goes to Frank Willett. And just as an aside, he came second last year, and I think that's a first for 2 years running. From the Neuroprosthetics Translational Lab, co-directed by Jamie Henderson and Christian Shenoy, for his paper, co-authored with Donald Alvancino, published in Nature. In the study, the authors developed a brain-computer interface for typing that could eventually let people with paralysis communicate at the speed of their own thoughts. The work involved repurposing a machine learning algorithm originally developed for speech recognition, which allowed them to estimate characters based on the neural activity of the user alone. They then improved the accuracy of the algorithm from 94 to 99% by including predictive language models like those driving your autocorrect function on your smartphone, and so that the participant was able to type at a speed of 90 characters a minute, which was twice the speed of earlier invasive brain-computer interfaces. Just pretty remarkable. This work and the new approach will help the field entirely rethink how brain-computer interfaces can be designed. So very spectacular. And just as a final note, the paper received the cover of Nature, a Nature News and Views article. It was covered by several news outlets. Over 4,000 news articles resulted from it, including segments on NPR, the BBC, Science, CNN, and these are just to name a few. So it got a great deal of attention, as it deserved. All of this work that I've covered is spectacular. And I think there are a lot of people up on the stage already, but for those of you who are only receiving this online, here are the mugshots of all the winners, and congratulations to everyone.
Congratulations, you guys. Just fantastic work. Absolutely fantastic.